Yeah, so look, this is the first time that we have actually validated the efficacy of RG-007 in an independent laboratory. So the bulk of our data in terms of how well the drug works in our preclinical studies has been done in the lab of our Chief Scientific Officer, Professor Bruno Maloney at Perrin Institute. And of course, we have absolute confidence in his scientific ability. However, really, we wanted to put the drug in the hands of an independent clinical research organisation and use their really well validated model and say, look, here is the drug. Please test a whole range of different doses, and also get the pharmacokinetic data from this study as well, which tells us a lot about the way that the drug is moving around the body, when it sort of peaks in terms of its concentration, how long it stays in the body as well. And so really was the first time that we have independently validated it. And the results that we got were actually phenomenal. The CRO even themselves commented just how exceptional the results were. So it not only gives us the confidence that we know that this drug works, but it gives us now a lot more data to understand exactly where it's working in terms of that dose level, how the PK data matches up with what we're seeing now in our phase 2 trial in terms of our actual stroke patients. And it will give us just a lot more information now to make sure that we take the right dose into the next trial design for RG-007 in stroke patients. So with this new preclinical data, as well as both our phase 1 data in terms of the pharmacokinetics, as well as our phase 2 data now, which we have in terms of the pharmacokinetics, it just gives us a much richer data set in which to design the next trial. So I often tell people we are in such a better position post this phase 2 with all this data coming in now than we were at the beginning of the phase 2, and we're really starting to have a really clear picture of where the drug is working, at what dose it's going to work best, so that we can really hone in on the phase 2B study, to really make sure that we are now, really going to emphasize that efficacy signal. So combine it all together, we feel that we are in a really good position to design the right trial the next time. Yeah, so the next part of data that we're waiting on, which will really complete the picture, is a report from Brainomics. Brainomics is a spin-out company from Oxford University. They have an FDA-approved AI software tool, which allows us to get a lot more granular detail in terms of the types of patients that came into our phase 2 study, the types of brain injury that they had not only when they came in, but also post-placebo or RG-007 treatment. So that really is the last piece of the puzzle. So we'll combine all the data that we have and, like I said, really hone in on the right trial design. The point of the phase 2 was very much, signal searching. We did, throw the net quite wide. We wanted to bring in all large vessel occlusion patients to that trial and really understand, does our drug work best in a certain patient subgroup? So once we get that Brainomics data, along with this current MCAO modelling, as well as all the PK and other data from phase 1 and phase 2, it will start to paint a really clear picture. And it just allows us to, as I said, really hone in on where the drug is working best, and we amplify that signal in the phase 2B study.